Competence.
All right, take it away.
Thank you so much. Okay, so I've already introduced myself, but the project we're talking about today is looking at the impact of antibiotic use in infancy on asthma and allergy incidence in 600,000 Canadian children. And this is across the two provinces of BC and Manitoba. Next slide, please. So I have no conflicts of interest. Next slide. So I want to respectfully acknowledge that we live and work on the traditional unceded territory of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam nations. This is a picture of Port Moody, where I live. But I also want to recognize that data for our cohort studies were collected on the traditional unceded territories of all First Nations across BC, as well as from Treaty 1 territory and the traditional homeland of the Red River Métis in Manitoba. Next slide, please. So I kind of already covered our stewardship program, so we don't need to go through that anymore. So next slide, please. So these are our data disclaimers. I won't go into this, but just to mention that all data from BC were provided through the Population Data BC platform, and all data from Manitoba were provided by the Manitoba Centre for Health Policy. Next slide, please. So just a bit of background. So why are we talking about antibiotic use in infancy and allergic disease outcomes? So the kind of proposed mechanism of these two things being connected is through changes to the infant gut microbiome, particularly very early in life, in the kind of first year or even in the first 6 months. So we know that there are prenatal, intrapartum, and postnatal influences on the initial seeding and subsequent colonization of the infant gut microbiome, which has impacts on metabolic pathway development and on immune system training. We also know that disruptions to a healthy gut during these crucial early days of immunological development can have lasting impacts on chronic disease outcomes. Of particular interest to us are the outcomes of asthma and other atopic diseases. So in the first 6 months of life in particular, some key influencing factors, both in utero and postpartum, are things such as preterm birth, mode of delivery, how baby was fed, as well as antibiotic exposure, which we know can have a damaging effect, not just on pathogenic bacteria, but also on commensals as well. And this has a lot of impacts on the ecology of the microbiome, particularly in reducing diversity and the loss of bifidobacterium species, which play a key role by producing short-chain fatty acids. And those short-chain fatty acids play a key role in immunological development. Essentially, Th2 cell production is upregulated and T regulatory cell production is downregulated, which taken all together can lead the immune response to be kind of hyperinflammatory later in life, later in childhood, which is why we're kind of talking about antibiotic exposure and atopic disease outcomes in a nutshell. Next slide, please. So this is not the first time that this question has been investigated. Sorry, my font's very strange on your computer, but that's fine. We're going to go with it. This is from a systematic review and meta-analysis conducted by our colleagues in Switzerland, and they looked at all sorts of different chronic disease outcomes. But of particular interest to us was obviously the atopic diseases. So they found in 51 studies, across 51 studies, that children who were antibiotic exposed in their first year of life were at over 2 times the adjusted odds of developing asthma. They also found significantly higher odds of developing other allergic diseases as well, such as eczema, allergic rhinitis, wheezing more generally, as well as even food allergy. And they found that exposure in the first 3 months of life appeared to be the most important or confer the strongest effects. Next slide, please. Another interesting layer in this kind of picture is the role that breast milk plays. So this is from work conducted by our colleagues in the child study, which is a national prospective birth cohort looking at early life influences of childhood atopic disease. And they found that infants who were antibiotic exposed and were formula fed were at over 3 times the increased risk of childhood asthma compared to antibiotic unexposed infants. Whereas if a child was exposed to antibiotics but were breastfed at the same time, they were at no higher adjusted odds of developing asthma compared to antibiotic unexposed infants. So essentially what that means is that it's not the breast milk itself that appears to be protective against asthma on its own. It appears to be replenishing a disturbed gut microbial environment caused by the antibiotics. And this is through the presence of human milk oligosaccharides, which are special sugars in breast milk that feed the bacteria in the baby's gut, not necessarily the baby themselves, which supports the growth of, in particular, bifidobacterium, which then contributes to this healthy immunological development. Okay, next slide, please. So this was from work in our group in collaboration with the child study from a few years ago. And we started off by looking at the kind of ecological picture in BC. So on the left-hand side, in the blue line, we see a 77% reduction in antibiotic use in babies between 2000 and 2019. I can tell you that this is still going down at a less steep rate, but it's still declining. I can also tell you that we haven't actually seen a meaningful increase in invasive secondary bacterial infection rates for our kind of key infection indicators in babies over this time period. So this appears to have not conferred a kind of infectious disease cost, if that makes sense. It appears to be a relatively safe reduction, even though it's so dramatic. And then in the red line, we see asthma incidence in children aged 1 to their 5th birthday. And that declined by 44% over the same time period. So this really piqued our interest. And then as part of this same paper, this collaboration with a child study, they looked at about 2,000 kids in BC and found that after adjusting for patient level factors in this smaller group of children, antibiotic exposed kids were over 2 times increased odds of developing asthma in childhood. Next slide, please. So this then led us to want to investigate this question in individual children so that we could adjust for individual factors, but scaled across entire provincial populations using administrative data. So what we did was we conducted a retrospective cohort study, 2 concurrent retrospective cohort studies in BC and Manitoba, recruiting all infants born in these 2 provinces between the years of 2001 and 2011. We followed all the kids up to age 7 to confirm atopic disease trajectory. And we also built in a parental look back period of 5 years before each birth to ascertain mom and dad's atopic disease history. So we could build in that kind of genetic predisposition as well. So our entire data set was 1996 to 2018. Our exposure was antibiotics before 1st birthday, and our outcomes were asthma, eczema, or allergic rhinitis before the 5th birthday. We performed multivariable logistic regression models, and we also decided to cluster the standard errors on the mother's ID because moms could contribute more than 1 singleton birth to the cohort. And so we wanted to recognize the fact that children raised within the same family, within the same home environment, would have similar early life exposures, and they should therefore be treated as such. We also performed group-based trajectory modeling to identify different trajectories of atopic conditions over time. I'll talk more about that later. And then we use multiple imputation to kind of deal with missingness. If you're interested in our methods, we did publish our study protocol in BMJ Open a year and a half ago, so that can be found there. Next slide, please. So I won't go into this in too much detail, but apparently this was our conceptual framework, essentially. So this is a directed acyclic graph, and it's simply to recognize how complex this pathway is, and that antibiotics are by no means the only influencing factor along this pathway. So baby antibiotics, our exposure of interest is on the left-hand side with a little triangle symbol, moving through kind of the baby microbiome, changes to the baby microbiome, moving through a pro-allergic immune skewing, and then lastly to our outcome of interest, so allergic outcomes, but also taking into account all of these other different factors. So factors related to the birth event, to kind of clinical visits, to the parents' genetic history, socio-demographic factors, environmental factors. There's all sorts of things at play here. And we essentially built this DAG in order to help us build our models so that we could make sure that we didn't have any open backdoor pathways for potential confounding. And it was really helpful in building our models, essentially. In order to capture all of these different factors, we pulled data from multiple different administrative data sets in BC. And as I said before, they were provided by Pop Data BC. And then we matched them as best we could to equivalent Manitoban data sets. And one of the reasons why we performed this study in these two provinces was because the data were very comparable between the two provinces, which is unusual. Okay, next slide, please. So let's do some results. So we had several exclusion criteria that we applied. So on the left-hand side is BC, on the right is Manitoba. So we started off in BC recruiting over 464,000 infants. And in Manitoba, we recruited almost 142,000 infants. I say recruited, we collected data for, for almost 142,000 infants. And after applying our exclusion criteria, so excluding children who were part of a multiple birth, who were placed for adoption within 26 days of birth, who were born prematurely, or who did not have full follow-up to the age of 7, we ended up with 342,686 included infants in BC and 133,383 included infants in Manitoba. Next slide, please. So in BC, 36% of our infants received antibiotics before their first birthday. The rate of antibiotic use in infancy declined over time, which we would expect based on our surveillance data. And the most commonly used antibiotic across all years of the study was in the blue bar there, beta-lactam penicillin. So that's essentially amoxicillin. About 9.5% of our kids were diagnosed with asthma before the 5th birthday, with confirmation before the 7th birthday. And then under 1% were diagnosed with eczema or allergic rhinitis. This is almost certainly a gross underestimate, because all parents will know you don't usually take your kids to the doctor for a mild rash or, a runny nose and sneezing, but we're capturing the most severe cases here. And then this graph on the right-hand side is our asthma diagnosis rates, which also declined over the study period. And asthma was much more likely to be diagnosed in boys than in girls, which we would expect. Next slide, please. In Manitoba, 43% of our infants were given antibiotics in their first year. Again, this rate declined over time. Again, amoxicillin was the most commonly used antibiotic for all years of the study. About 10.5% of the kids were diagnosed with asthma, and over 5% were diagnosed with allergic rhinitis. We weren't able to assess eczema in Manitoba simply because of how the codes are extracted. We couldn't get a specific enough code for eczema, so we could only look at that outcome in BC. And then this graph on the right-hand side is, again, asthma rates in childhood, again, declining over time, again, much more likely in boys in the orange line than in girls. Okay, next slide, please. So these are our adjusted models in BC. And these were all of the factors that I touched on before that we ended up adjusting for. At the very top in the red star is antibiotic use in the first year, our exposure of interest. And this plot on the left-hand side is our asthma outcome. So after adjusting for all included covariates, antibiotic exposed infants were at 1.64 times higher adjusted odds of developing asthma compared to antibiotic unexposed infants. Sorry, the font has gone a bit crazy on that one. So yeah, 1.64 times higher adjusted odds for asthma. On the left-hand side is eczema. And after adjusting for all of our covariates, antibiotic exposed infants were at 1.17 times higher adjusted odds of developing eczema than unexposed infants. Next slide, please. And they were at 1.42 times higher adjusted odds of developing allergic rhinitis compared to antibiotic unexposed infants. Next slide, please. For Manitoba, they had fewer covariates to adjust for because they had more missingness, but just to assure you, we reran these models using imputed data, using the full covariate set, and the outcomes were basically no different. So on the left-hand side, we have asthma. On the right-hand side, we have allergic rhinitis. So after adjusting for covariates, antibiotic exposed infants were at 1.55 times higher adjusted odds of developing asthma and 1.42 times higher adjusted odds of developing allergic rhinitis compared to unexposed infants. Next slide, please. So essentially, we're seeing quite a strong clinically and statistically significant signal across all outcomes in both provinces so far, particularly so for asthma. But we wanted to look at our exposure in a bit more detail. So we wanted to investigate antibiotic quantity, spectrum, and class on our asthma outcome. So on the left-hand side, you have BC. On the right-hand side, you have Manitoba. All of these models are adjusted for all covariates in the previous models, but we just changed the exposure variables. So our top model, we're looking at the number of courses of antibiotics babies received in their first year. And essentially, we can see a kind of dose-response relationship. So the more antibiotics the babies received, the higher adjusted odds of developing asthma in both BC and in Manitoba. The second model is looking at narrow versus broad spectrum antibiotics and what the babies were exposed to. We see slightly higher adjusted odds in babies who only received a broad spectrum antibiotic as opposed to a narrow in both provinces. And the last model is looking at specific classes. And interestingly, we see that macrolides appear to confer the highest risk of developing asthma in both provinces as well, after adjusting for everything else. And this is something we're going to be investigating a bit further because we're starting to see that coming through in the literature in smaller studies very recently. So we're going to try and investigate why that might be. Okay, next slide, please. So we conducted a few sensitivity analyses because we didn't want to just leave it there. There's a lot of, as I said, it's a complex question. It deserves a complex response. So this is our conceptual framework for our our conceptual map for our sensitivity analyses. So we have our main cohort at the top there, off of which we ran all of our previous models that I just covered. Next slide, please. And we then we wanted to tease apart what indications the kids were receiving antibiotics for, to try and get around this common criticism that it's not the effect of the antibiotic, it's the effect of, say, the respiratory tract infection that the antibiotic is being used to treat. And it's the RTI that is predisposing a child to develop asthma later in life. So we restricted our cohort to only look at children who received antibiotics for non-respiratory infection indications. So they had no record of an RTI in their first year. And we still found a very strong signal in terms of higher adjusted odds of developing asthma in those antibiotic exposed kids compared to unexposed kids in both provinces. Okay, next slide, please. And again, so for our second sensitivity analysis, we looked at only infants who had at least one respiratory infection in their first year. And then we tried to tease apart who did and didn't receive antibiotics. And again, we still saw a very strong adjusted odds ratio, elevated risk of developing asthma in antibiotic exposed infants in this kind of subgroup. Okay, next slide, please. And again, so we also wanted to investigate, sorry, let's cut it off. I'll just kind of say verbally. So we also wanted to investigate the timing of antibiotic exposure, because as I mentioned before in the literature, we've seen that earlier antibiotic use appears to have a stronger effect on asthma risk. And so we kind of split the first year up into quarters and looked at when the baby first received their first antibiotic. And essentially we found in both BC and Manitoba that kids who received antibiotics in their kind of 0 to 3 month time period or 4 to 6 month time period had higher adjusted odds of developing asthma compared to those who received their first antibiotic later in their first year. And that was again in both provinces. So we're seeing that kind of consistent effect that we see elsewhere in the literature. Okay, next slide, please. We also wanted to counter this idea of reverse causation. So looking at, if infants had wheeze-related conditions before they'd even received antibiotics in their first year. So we excluded infants with a record of viral wheeze, recurrent wheeze, or bronchiolitis, or asthma itself, even though that shouldn't really be diagnosed in early infancy, but it did show up. So we excluded infants with any of those diagnostic codes in their first year, and then reran the models. And again, we still saw quite a consistent elevated risk of asthma in both provinces in antibiotic exposed children. And then lastly, for the fifth sensitivity analysis, we wanted to remove transient cases of atopic disease from the case group so that we were only looking at kids who went on to develop chronic asthma. Next slide, please. So this is where we performed group-based trajectory modeling to try and identify those different trajectories of chronic disease. And at the top here, we have BC, at the bottom we have Manitoba. And you can see in both provinces, 3 very kind of distinct trajectories of asthma. So on the bottom, you have no asthma. And then in the kind of light gray dotted line, you have your kind of transient cases in both provinces that are diagnosed early, but then they don't revisit for asthma later in childhood. Later in childhood before the 5th birthday. And that's the same in Manitoba. And then in your black line, you have what we call our early onset and chronic asthma group. So essentially we removed the kids in the light gray group, in the transient group, from our cases, and then we reran the models. Next slide, please. And again, we saw very similar findings to our main models. So higher adjusted odds of asthma in the antibiotic exposed kids. Okay, so that's it for results. So we'll just talk about some limitations of this study. So next slide, please. So some of the main limitations are that the outcomes were based on clinical diagnostic codes. So as I mentioned before, eczema and hay fever were likely an underestimate, but we are capturing the most severe cases. There's also a potential for misclassification of asthma, possibly explaining reduced associations compared with prospective birth cohorts in BC. So where previously in the child study we saw a 2 times higher adjusted odds of asthma, in our study we saw, kind of 1.65 higher adjusted odds, for example. So again, this might be a conservative estimate. We also did not have data for all the variables in our theoretical framework. So we didn't have data for kind of pet ownership, daycare attendance, or the mother's microbiome. But we did the best with what we could. And also pets and daycare don't appear to have very strong associations with the outcomes in the literature in smaller studies. But there is a potential for residual confounding. And then lastly, we were only able to capture antibiotic prescribing in the community after baby was born. So we had, whether baby received antibiotics during their hospital stay, during that initial birth event. But once they were discharged home, we only captured antibiotic use in the community, so through their GP clinic. But what we know from the literature is that this is the vast majority of antibiotic use in this demographic group. Okay, next slide, please. So in summary, we saw 64%, 17%, and 42% higher adjusted odds of asthma, eczema, and allergic rhinitis in antibiotic-exposed infants in BC, and a 55% and 42% higher adjusted odds of asthma and allergic rhinitis in antibiotic-exposed infants in Manitoba. The association with asthma was robust in children only receiving antibiotics for non-respiratory infections. We also found a dose-dependent relationship between antibiotics and asthma risk. So the more antibiotics the child received, the higher their risk of asthma. And we also found that macrolides appear to confer the highest risk in comparison to beta-lactam penicillins. So overall, the associations with all atopic outcomes supports our hypothesis that the mechanistic pathway is immune regulated and influenced by gut microbial dysbiosis early in life. So just briefly, one slide on the kind of KT work we've been doing. So our key audiences are varied, as you might imagine, and they are most importantly parents and soon-to-be parents. Researchers, clinicians, and policymakers. We currently are finishing up a research topic hosted by Frontiers in Allergy, Frontiers in Pediatrics, and Frontiers in Public Health. It currently has 12 accepted articles, 1 under review. And this is titled Preventing Childhood Asthma, the Neglected Impact of Existing Public Health Interventions. This will be published soon as an ebook. And we're hoping that this will serve as a kind of comprehensive volume on the subject to be referred to. We also put together a stakeholder advisory committee made up of all of our different key audience groups with kind of representation from all health authorities across BC, and they have been helping to guide our knowledge translation strategy. We've also been working with researchers at UBC to try and attribute asthma and allergy-related cost savings to antimicrobial stewardship in the community. So if we can start to tack on cost savings related to chronic disease outcomes onto antibiotic stewardship, that could boost the argument for investment in stewardship in the community. We have had presences at multiple conferences, and we are currently conducting outreach to our BC perinatal program colleagues and have received funding to produce some parental video resources as well. Okay, so this is to thank the team, our last slide. So huge thank you to the whole team. David Patrick's our PI in BC, Nathan Nickel is our PI in Manitoba. Max and Lisa in particular are incredible data analysts, biostatisticians who have been wading through these data sets with me for years now. So huge thank you to them. And huge thank you to our funders, CIHR for the analytical work and Pacific Public Health Foundation for the KT work. And that is everything. We have a few minutes for questions. So yes, we've whistled through this.
Thank you so much. Let me just stop the recording.